We are good to go.
Thank you. Hello, everybody. I think we can give a couple of more minutes for more attendees to join in. People is getting in to the Zoom link at the moment, so we'll give a couple of minutes. Well, I think that today is going to be a very interesting conversation because I guess that any project, regardless is neuroscience, cancer, cardiovascular, plant biology, doesn't matter. Any project doing single cell technologies will have to go through the same process of asking, how many cells, how do I handle the samples, what is the multiplexing capacity, what I can do, what is the best strategy, and basically all the pre-design of the experiment, which is, I guess, the most important part of it, is just making sure that to get the best out of the technology. So today is going to be a lot of discussing these aspects, not specifically in any topic, not specifically applied to probably all the projects that anybody could think about. So I think that this is going to be a very interesting, and then because we don't have an invited speaker, we have a lot more time to answer questions, and then, perhaps it could be a lot more interactive in asking questions. Certainly I have a lot of questions I would like to discuss with Yvonne. So I think this would be a very interesting opportunity to discuss these questions. So, well, now today is the 6th seminar, so I'm very happy to we actually made it to the 6th one. So we've come along a long way through all the lockdowns and all the restrictions and everything, but I think that it was a very good idea to actually initiate this seminar series. Even they're virtual. The idea was to have them face to face, but that's not possible, as we all know. This is the new reality. But also we had the opportunity to have Yvonne consistently. She's based in Melbourne, but we have the opportunity to have her consistently here through Zoom, which is one of the the very few good things about this COVID pandemic is that we are learning different ways to communicate and different ways to have sort of this kind of communication. So that's great. Anyway, so I think that we can start. It's 4 minutes past 10. So we can start while everybody else is connecting. Again, it's a pleasure for me to introduce and welcome Yvonne again to the 6th seminar series. Yvonne is the Science and Technology Advisor for 10x Genomics, and she has a wealth of expertise in genomics. She's worked before getting into 10x. She's working for many other companies and many other acquiring different expertise in different aspects of genomics. So she comes with a wealth of information. So she's very someone to basically ask for any information about not only 10x, but in terms of design experiments and approaches and things like that. So today she's going to be talking about a little bit of tips and tricks for single cell and spatial gene expression. And then we have a Q&A for everybody. Everybody welcome to discuss and switch on your camera or whatever, or put it in the chat. And this is meant to be an interactive session. So anybody interested in starting experiments or interested, they're already using it, but they have questions about how to scale up or whatever, I think that will be a perfect topic for discussion today. So Yvonne, all over to you. Thank you very much for being here today.
Thank you, David, for having me and for the introduction, and good morning, everyone who's joined. I have, a number of slides today. I think this topic is actually, we could basically talk for days on this, diving into each application and specific sample type, etc. But I think I've picked a few things that we get asked very often to discuss. And as David said, please just at any point, if you have a question, just jump in, and we keep that quite quite open today, as it's designed, you know, specifically to give you some ideas on how to move forward into the next single cell experiment that you might be planning or getting started for the first time with a single cell. And I know David has the single cell capacity in his lab. I will have a couple of slides on spatial gene expression as well to just get everyone sort of started thinking about it as well. So we'll just cover a few of these experimental considerations, logistics, experimental design, and the grant resources we can probably leave off at the end. And just if anyone does want support with this, we actually have a number of resources online to help with grant applications as well. So just as a brief summary, David actually said it brilliantly, just as an introduction. When you get into a single cell experiment, it is crucial to know exactly what your research goal is, because that will define your experimental plan. It will define, what input material you can use or you should use, whether it's cells or nuclei. And it will define how many of each you should be planning on using to get the answers you want. So there's a number of things to consider, whether you characterize and identify heterogeneous cell populations, or you want to discover new cell markers, regulatory pathways, uncover novel cell types, cell states, and rare cell types that will all define which assay to choose. So when you think about a new single cell sequencing experiment, there is a few parts to the equation that need to be considered. So sample quality and sample preparation is the key. And I think if the few of you that may have been in our very first conversation, our very first talk that we had here in the seminar series, where Luciano presented and said that it's basically probably about 80–85% of the whole experiment comes with your sample and with the sample quality and cell viability. So that is a big component that needs to be thought about. And what we need to look at here is that we maintain the single cell behavior and disturb it as little as possible. And what that means is what you're looking for is a high quality sample with very high cell viability. You'll see in our documentation that we go 70% or more. That's said. I want to highlight though that 70% is the bare minimum. We would actually always recommend a higher viability percentages, ideally 90% or more. If you can, get your sample quality or sample preparation optimized to achieve that kind of cell viability that is desired, rather than on the 70% line. Then the sample should be clean, and you want to minimize cell stress, but we go into all this in a little bit more detail. And then we all know that in a single cell experiment, logistics are really important. So it is important on the actual sample prep, library prep, and sequencing data analysis side of things, but it is also mainly considering how will I get my sample from A to B, and how do I make sure the cells stay viable until I actually load it onto my Chromium controller. So that does require a plan. We often say it in leading up to a single cell experiment, and in your case, having the facility at UTS available, I think that logistics part is made easier for you. However, it still does require careful planning in terms of the actual time it takes. Not only for your cell suspension preparation or nuclear suspension preparation, but once that's done, that the assay has to be loaded within about 30 minutes of your samples being ready. So that needs to be considered. And then, of course, your experimental design, what you're actually trying to find, and having a plan is, I guess, the best advice that we can give. have conversations with us or with David and the team when you are ready to to undertake an experiment, because we can add you know, additional information to your specific project. And something that is always also highly recommended by us is, if you're not quite sure if it's something where you don't have an example in the publication of something similar being done, it's always a good idea to run a very small pilot experiment with either just a few samples on a standard gene expression assay, for example, but we also have a kit that's called the Low Throughput Kit that profiles just about 1,000 cells. It operates on the same 8-cell format, on the same 8-well chip that we have, but it just runs a lot less cells, so it is also significantly cheaper. And it is a consideration you could have to just run a test with your experimental setup. So you would have seen this slide a few times, the different capabilities we have with our portfolio, where you can look at, you know, just standard gene expression, looking at our polyadenylated mRNA molecules. But if you're looking at, you know, T and B cell receptor information, you would use our immune profiling solution, where you could also pair that with the 5' gene expression. If you are looking at specifically, so here you can pair that with cell surface protein, antigen specificity, and the full-length TCR, BCR receptor information. But if you're still studying cell differentiation, cell lineage mapping, you would probably use our ATAC-seq epigenomics assay, or our multi-ohm assay that's combined ATAC-seq with the gene expression assay, for example. But if you're also looking at harnessing the power of CRISPR screening, CRISPR perturbation screening, this is also enabled with our technology. And depending on what it is you want to do, you'll have to decide what sample type or input material is required. So if you are looking at protein plus RNA, so if you're looking at cell surface protein and a combination of gene expression analysis, you'll need to use whole cells. If you're only looking at gene expression on its own, you can obtain this data either from whole cells or nuclei. But for the assay of looking at chromatin accessibility, so open chromatin, for that we are actually limited to just using nuclei. And something that we always highlight, and if you've attended one of our sessions that our field application specialists run, when they talk about sample quality, it is absolutely crucial that the quality is high. And what we mean by that is that the cell needs to be intact, so the cell membranes need to be intact, and nucleic acids are maintained inside the cell, and that will result in very little background signal and clean single cell data, because you can very accurately map every single transcript that's being captured back to the cell that it came from. And then if you have cells that are compromised, so that means the cell membranes are disintegrated slightly or compromised in any way, RNA starts leaking out. And when that happens, and if it's only with a small subset of cells in your specific sample, that increases your background signal actually quite dramatically and leads to poor single cell data. You'll still be able to get information out of an experiment like this with compromised cells, so that is actually quite limited in the information you get out. So we at this point, when you're starting and assessing your sample, and I have a few more slides coming up, it's very important that the cells are as intact as possible. And going back into this a bit more detail, what makes a good quality sample? What does it mean we need an intact cell? So we do want to absolutely reduce the physical decomposition of the sample to prevent this RNA leakage from the cell, the background, because it does increase the background, as I just said. And we also want to minimize RNA degradation. So you need to work fast, you need to work on ice as much as possible when you are generating these single cell or nuclei suspensions. And we want the cell suspension or nuclei suspension to be clean. So here's an example on the left what this looks like. And it means there is very little cell aggregates or clumps that are visible. So here it's a very nicely separated cell suspension that we can see. We don't want to have any or very, very little subcellular debris and free-floating RNA DNA, which obviously you can't see in this in a microscopic image. But if you have the intact cell suspensions without clumps and debris, you will be having a very clean and intact sample type. So here is again on the left the cell viability bigger than 70% or greater than 70% that I mentioned, and it is definitely desired to go way higher than 70%, ideally 90% above. What we also want to clarify, I want to make sure a good quality sample is characterized of, is that the cells are healthy. So we want to minimize any biological decomposition, again, that can lead to RNA leakage and RNA degradation. And this is sort of done with assays where you have a live dead cell stain and you observe and count the number of cells that are viable and healthy versus the number of cells that have died. And if you have large numbers of dead cells, there is ways of removing those from your sample, and it's advised to do so if you know, if you are way beyond a few dead cells within your sample. So on this table here, we have summarized just a few differences between using cells or using nuclei, and most of them are quite obvious when you look at them. It's just important to keep in mind when you're using nuclei, you will not be able to obtain cell surface protein information or VDJ information, but you will be able to get a gene expression and epigenetic gene expression information just as much as you get this from cells. You can only acquire a cell suspension from fresh tissue, for example, that can easily be dissociated. Once you have actually fresh flesh frozen tissue without dissociating prior, or tissue that is very difficult to dissociate, in those cases, it's most often only possible to extract nuclei from this sample type. And that means, you know, so you are restricted in a way in the analytes that you can look at. So this is an important thing to consider when you assess your sample, the material that you are getting. And with cells, you are able to look at spliced mRNA and cell surface protein, as I said, but from nuclei, you will also get unspliced RNA with lots of introns in the information, as well as chromatin. But as I mentioned, no cell surface protein. So maybe another thing that I point out again, so if you are freezing tissue to extract nuclei later on, so that would be a way to be able to transport tissues to get processed for a single cell experiment. Before I get into a few more sample prep considerations, I want to stay on the nuclei quality and assessing this, as this is also often asked by a lot of the users. How would I assess my nuclei, and how do I decide this is okay to proceed or this is not a good quality nuclei sample? So on the left is one part of assessing nuclei quality, and that is assessing the efficiency of the cell lysis. So leading up to a single cell experiment, you typically perform a lysis time course. to identify the most optimal lysis time. And you do this by looking at, for example, a count test type instrument where you perform live dead cell stains, and you see, you know, how many cells are present. So what you want to see is very few live cells, and the nuclei stain as dead. And then you want to clean clump-free nuclear suspension. And suboptimal lysis, as an example, would look like this, where you still have a lot of live cells present in your sample, and you can see this here in the percentages. So again, please feel free to contact us when you are in this state and you want to decide whether to go ahead and optimize lysis, or whether you can. This is considered contact lysing conditions already. The second part that needs to be assessed is the actual quality of the nuclear membrane, and you do this on a microscope. So typically, you know, 60% magnification in a bright field microscope. And what we want to look for is what you see in pictures A and B. You want to see a smooth, contiguous membrane with very little or no blebbing observed. And when you see images of your nuclei, like represented in the pictures C and D, the nuclear membrane here is compromised. We see a lot of blebbing and basically almost destroyed nuclei. And at this point, we would say this is a very poor quality nuclei. It wouldn't advise to go ahead with a sample type like that. Now, we always, you know, everyone is trying their best to get sample prep optimized and to get the best type of quality of sample, but there is still in many cases the possibility that your sample doesn't meet necessarily all the criteria that we have for a single cell experiment in order to go ahead. And there are still things that you can do. So if you have cellular nuclei isolation and sample prep and enrichment, you perform sample separation, there is specific enrichment method that you could try in order to remove dead cells from your sample type, for example. So you can just simply try additional washing steps to get rid of those of debris, for example, or you can enrich for live cells. There is products available to do this, to clean up your cellular debris. You can remove ambient RNA. There's also protocols available to do this. And then there's also the option to enrich or deplete for a very specific cell type of interest via FACS sorting, for example. So if you're only interested in a specific cell type, looking for a subpopulation, we do recommend doing a FACS sorting step and increasing the number of cells that are desired for analysis, and removing the undesired cell types. And that, of course, is only possible if there is an antibody available to stain very specifically for this kind of cell type that you're interested in. Before moving on to the sample prep workflow, maybe I'll just pause quickly and see if there's any questions.
I might just comment a little bit on that sort of a sample prep, how important the sample quality is in terms of, because I think one of the main questions that many users have is my sample suitable? You know, people having a biobank of archived tissues or cells, and, you know, they wanted to jump into the technology. So, I mean, I can't stress how important is having a proper sample. And, you know, if the users are already expert in getting single cell suspensions for other techniques, flow cytometry or whatever, then you are already like 80% into a proper single cell RNA-seq experiment. If you never dissociated your tissue of interest, then that is when you need to start really at the beginning to optimize that, to make sure that what Yvonne has been saying really like tick all the boxes on that particular one. And then from there you can think about, you know, scales and stuff like that. But the sample prep, as any other experiment in biology, is the most important part of any downstream application, whatever that might be.
Yeah, and I just want to mention that we have a lot of demonstrated protocols available for a large variety of tissue types and sample types as input material. We do appreciate that it doesn't always cover necessarily a sample type that you want to work with. So please, in any case, reach out to us. We can always contact R&D, we can contact tech support, and draw on the experience that we have with customers as well that are using these sample types and using, you know, modified protocols or user demonstrated protocols as well that we can provide to you as well. So please, in any case, reach out so we can provide as much as information and protocols as possible to you when you start. I don't know what's going on here, sorry about that. When you start your optimization. So this is the workflow as you would have seen this now many times. But once we have actually made the libraries, there is no time pressure as such anymore at all. So once you've got this as libraries, you can freeze them, you can take them to sequencing straight away and analyze in your own time. So we'll just keep focusing on the actual sample collection side of things and how cryopreservation and shipment affects your cells. And on this example here, I know PBMCs is a very common sample type, and it's fairly straightforward to isolate, but I'm just showing here that the correlation between fresh and cryopreserved samples when you look at gene expression is extremely high. So it performs very similarly, and this is based on using the protocol that we have available for this type, isolating from blood, obtaining PBMCs. So just want to highlight again, we have a lot of these protocols on our support website. And if any of you have used them and they haven't worked, or have used them and you know, found them to be really easy to follow, would be also very interesting to have that feedback. So then we know that specifically for tissue collection and transport, there is many different ways of doing this, and there's only a certain types of tissue that you can obtain sometimes. If you have absolutely no influence on how the tissue is collected at a surgery, for example, from a patient, and all they do is they snap freeze it straight away, and you have no way of changing that kind of sample type, then in this chart here, I just want to highlight there is very limited ways of how you can move forward, and the only thing that you can do then is extract nuclei. But there's also the possibility of getting the tissue collected and putting it in a storage solution at 4 degrees, and at that point you will be able to dissociate the tissue and obtain cells or nuclei if you wanted, but usually, typically, people would go and opt for cell extraction. And you know, you can also take the tissue and directly dissociate it, or you can cryopreserve it following one of our specific protocols, and then that will also allow you to follow on with the tissue dissociation protocol to extract cells and perform, you know, downstream analysis in a single cell experiment. So there is some ways, and David just, you know, highlighted this. If you have access to a tissue bank, there is typically only a very specific way that these tissues have been preserved, and then from that following on, you'll consider which way or which type of sample, which type of nuclear cell you can extract going forward. But what it does is it does allow you to also think about storage timing. So from time of collection to time of preparation, to help you plan your experiment. So the whole time it's moved on its own, and now I can't advance. And then a very similar question and a collection method and when to use and what to consider when you collect blood, specifically where we recommend this is only validated tubes to be used, and we have sort of a decision tree where, again, it depends on where your blood comes from and if there is any way of isolating cells directly at the collection site, and what to do and what to follow, or whether there is no capability of this and the blood will basically be shipped at 4 degrees or room temperature for up to 48 hours until you must proceed with the extraction. So we say that the storage, at 4 degrees, again, minimizes the cell stress and also the T cell apoptosis within the blood sample. So it's quite important to at least have access to ice or cold packs in order to store the sample appropriately and transport it to your lab. Maybe on this one, if I mention the PBMCs we've talked about just before, but for neutrophils, for example, there is a special collection method that's recommended in order to collect these appropriately. So it's the Vacutainer tube only that we have validated, at least to be compatible with our technology. So you can store that at room temperature, but you have to process this within 24 hours. In order to appropriately preserve the neutrophils and extract them. So there's, again, the recommendation or consideration here is to process as soon as possible. You work with this one at room temperature, you actually don't put it on ice. So, compared to all the other methods that you're doing. So it's very important to know where your sample comes from, how it's being collected, so that you have an understanding of how much time you have until you have to process your sample. Something, and we just had this brief discussion before the seminar today started, we have a fixed RNA profiling kit coming that is supposed to make this, what we've just discussed, a bit easier, because this is a standalone kit that will allow you to fix your sample with the standard sort of 4% pyroform aldehyde solution to stabilize your sample for later analysis and basically take all these time constraints and difficulties in terms of capturing your sample, storing it appropriately, getting it transported out of the equation. So this is a standalone kit that we have coming in next quarter, so the beginning of the year. It will work with the probe sets, a little bit similar to our Visium FFPE probe set that we have. The probes itself are going to be slightly different, so we are adding in more probes than we have in the FFPE solution at this point, and it will be launched for human to start with. And that's then followed by mouse very soon after. Something that we often get asked when it comes to this particular kit is the multiplexing capability at launch is not going to be enabled straight away, and it's not going to be cellplex either. So it will have its own cell multiplexing capability that it will be made compatible with when we have it ready. And some preliminary data that we can show from 7 days of storage and how this compares in terms of UMI count per gene, the gene as well as cell type annotation, compared between the fixed, non-fixed, and 3 to 7 day time frame. So we are accumulating more and more data at the moment because we are anticipating for this kit to be able to fix your sample for processing for 3 to 6 months. So at launch or closer to launch, we'll have a lot more data available that we can show. But here is just another representation of the sensitivity of the 3' in the 3' workflow for this particular assay. So I think this is a good product. Once it comes out, it will be making life a lot easier. It doesn't quite have the same capabilities in terms of, it will be compatible with cell surface protein detection. You won't be able to do multi-omic, for example, with it at this point. So 3' gene expression, cell surface protein is going to be enabled. So if that's something that you are considering as an experiment, and you would have a cell type that's very difficult, a sample type that's very difficult to collect and transport, this kit could be a real good option to use as a preservation method. So next I have one slide for CMO labeling with CellPlex, where we've put together recommendations when using our lipid tag cell multiplexing oligo or CMO for a single cell experiment, because there's a number of things to consider. We don't recommend using CellPlex as a first-time user, as your first experiment. Ideally, you run the cells, your samples individually, and potentially as a downstream experiment, you can consider pooling samples together with the CMO tags. I think probably the most important consideration when using CellPlex is you have to actually have a large number of cells as your starting material available, because we are expecting about a 50% loss of cells or nuclei during the labeling and washing steps. So we recommend you start with at least 500,000 to 2 million cells if your sample is not limited to. So any limited sample is probably not suitable for a CellPlex experiment. There is some recommendations for cell pooling. We recommend equal pooling ratios for optimal multiplet detection. And there are recommendations if you do, go and perform non-equal pooling, and there are some good reasons why people do want to do that. We do recommend that at least each sample accounts for 5% of the total cells or nuclei in your pooling experiment, and that's for reasons that is needed for the cell ranger to detect your samples optimally. But something here that I want to highlight also is that we recommend pooling 3 to 4 samples together. That seems to be the sweet spot for CellPlex and for sort of number of cells that you can then capture per sample as well. So just also keep that in mind when thinking about CellPlex as a multiplexing technology. One thing we have for sample storage, we recommend using fresh cells when doing CellPlex. It performs quite well on fresh cells. It is less reliable or less stable when using frozen cells. So at this point, we would highly recommend to use fresh cells as an input material. Methanol fixed cells are also not supported with CMO labeling, and also quite important once you have actually labeled your sample that you don't cryopreserve those afterwards. And again, we recommend 90% cell viability for the labeling going into the experiment, and the ambient RNA should ideally not be present at all when you're doing CellPlex. We recommend cell counting of the cells or nuclei, so you can use both. CMO tags are compatible with either input material, but we recommend you count them before and after pooling. It's very important to perform accurate counting to enable correct representation of each sample. Any comments or questions around CellPlex?
I have a couple of comments, but I think that we can leave them until the end. I believe you're going to start into the spatial.
Just wanted to do spatial now, yeah, as some considerations.
Yes, that's great.
So here I've put together general guidelines depending on which workflow you're choosing, whether it's the Visium Fresh Frozen or Visium FFPE. So again, as a reminder, for Visium FFPE, we are actually using here the same slide, the same Visium slide, but we are incubating the sample with a large probe pool, whereas in the Visium Fresh Frozen, we are detecting these mRNA molecules from the tissue directly. And here, it's quite crucial to test your samples that you're planning on using to test the RNA quality, and we do this by obtaining the RIN value, and we are asking for a RIN value of at least 7 for tissue types to return high quality Visium data. The workflow is asking for a tissue permeabilization test, and that is critical to do that every single time you're changing sample type, for example. So whether it's just a different tumor type or a different organ, it is very important to go in and perform tissue permeabilization testing again to ensure that the timing is spot on for that particular sample type in order to avoid transcript diffusion into the neighboring cells. When you're doing immunofluorescence, it's important to stick to our protocols, especially if you've used the very same antibodies already in a standard immunofluorescence workflow. For the Visium Fresh Frozen, we do recommend to retitrate your antibodies and make sure that you have the correct concentrations for using it on these slides, because the incubation times are quite different, or will be most likely different to what you're used to. If something goes wrong, we get asked this a lot, we recommend practicing placing these sections onto a slide where you draw on these capture areas. However, you practice and everything went fine, but then when you actually perform your placing of the tissue sections onto the Visium slide and it goes wrong, it folds over, or something else is not quite right, there is actually a reset protocol available for the Visium Fresh Frozen workflow where you can remove the tissue section and start again. Another thing that's very important to consider is when you practice, as well as in the real workflow, we need to keep the fiducial frame clear as much as possible. It doesn't if a slight overlap of a tissue on parts of the fiducial frame occurs, that's okay, but the majority needs to remain free because it is used for alignment purposes of the software between the gene expression data and the imaging data. What's very important for the tissue preparation is because you can use fresh frozen or frozen tissue, but it is important that it's not directly frozen in liquid nitrogen, because it does result in images like this. There is a very specific protocol that we recommend how this freezing is done in an isopentane bath in a liquid nitrogen bath, for example. Very important to stick to that protocol to ensure tissues look intact, as you can see on this example. And we also have some guidelines for the OCT embedding itself, so that you can perform it to size. So it fits into the capture areas, but also how you would score a tissue that is bigger than our capture areas in order to put them on across several sections of the slide. And in contrast, our Visium FFPE gene expression solution, here we are actually doing our quality control of the RNA using the DV200 value. So we are assessing the percentage of RNA molecules that are longer than 200 nucleotides, typically via extracting RNA with the QIAGEN fixed RNA RNA kit, and then running that RNA on a bioanalyzer nanochip and just obtain that percentage. It's very straightforward assay to do, but very crucial, as we are seeing that if the value is 50% or higher, you get quite high quality data from Visium FFPE. In saying that, we do see actually still some very exciting data with DV200 values that are a lot lower than that. So in the 20s, in the mid to high 20s as well. So if you have tissue blocks that are, old or bad, poorer quality, you could still obtain data from that if you have no way of utilizing a different sample type. Same thing here, practice the sectioning and placing sections on the slide and follow the best practices there. For tissue adhesion, that's actually quite important to make sure the FFPE section sticks to the slide. We have removed our tissue adhesion test as a must in the workflow, so that's not required anymore unless there is troubleshooting to do, in which case we would use it again. But if you follow the best practices for tissue adhesion, you should be okay with your tissue on the slides going forward. And then lastly, I just wanted to go into some of the sequencing consideration. Now, again, that is one of the things where we can also talk for a long time in terms of assessing this, how many reads should I use, what makes sense, where do I achieve saturation, and more sequencing doesn't actually add more of the information to it. So there is a number of tools available also to assess this, and the guidelines that we have in our user guides for gene expression with 20,000 read pairs per cell or nuclei, for example, or for any feature barcoding, 5,000 read pairs per cell. That's always a good starting point, but it really does depend on your tissue type and how many transcripts are actually available or are actually present in this. And we have validated the single cell libraries on a number of sequences to give you that the recommendations that we have, and it's important to follow those as a starting point. So if, for example, you can ask David, as he has a lot of experience with this already. In some cases, it might make a lot more sense to actually go in with twice as many reads as possible, or as we recommend, or just stick to the 25,000 reads per sample or per cell that we recommend. But really does depend on the complexity. So if you have an understanding on this, and you are expecting low complexity, you can adjust the sequencing recommendations accordingly. And there is a trade-off between sequencing depth and duplicate reads. So more sequencing doesn't necessarily mean more genes. We probably all know that from our experience with next generation sequencing. So there is some tools available to assess this, and I guess this graph here is also highlighting where in your sequencing throughput where you want to be. And then data analysis, again, this is just one slide I put together. There is a whole lot of resources on our website, but we do realize that it is in a lot of cases for first-time users a bottleneck. Or, you know, how do I do this best? Which tools to use and how to go about it. So we are actually internally working on this on a continuing basis to come out with these guidelines for each of the different essays that we have, and actually walk a first-time user through all the different steps they can take in order to look at their data and get clustering information, for example, or how to recluster, how to pick new, or how to pick individual cells that you think are potentially a rare cell cluster, how you can annotate those and identify them. And then also what other third-party analysis tools are available that we know are used very frequently and very successfully on top of, you know, the analysis you can perform with Cell Ranger or Space Ranger, and what you can visualize in Loop Browser. So we are not as such partnering with any of these third-party tool providers, but we are getting a lot more experience than what our users do with these tools. So we can provide guidance and can point you into the right direction on that point as well. And we are also working on organizing and putting together a specific bioinformatic workshops that specifically, you know, guide you and teach you on utilizing these tools, typically on example data sets that we have already also preloaded on the website, but we could do this with one of your own data sets as well, if that helps. So I just have some frequently asked questions, but maybe we can open it to questions now and some further discussion from the audience.
Yeah, I will be very interested. Thank you, Yvonne, for the overview. I think you will cover every single step. Can you hear me okay? Because I have some background noise, as every Wednesday. Sorry for that. I think you cover really well every single sort of step where you can get some issues. As this is a long process, starting from cell preparation to sequencing. So I'll be interested if anybody in the audience that is planning to do a single cell experiment in the future, or is doing it, or if they have any question about their own specific case, you can either put it in the chat, or you can just, I guess, unmute yourself and speak. In the meantime, I'll really like to perhaps one of the things that I am doing in terms of getting or increasing capabilities on the single cell facility at UTS is to start getting some of the tools that are necessary for the spatial transcriptomics. So there is a site assist machine coming up next year, I think, in the market, which I have my eye on, and it will be very interesting because this is one of the systems to, I guess, to streamline the starting point of the spatial transcriptomics, which is basically putting the tissue in the slide to make sure that there is a proper transfer of the tissue. And if it is an automated way to do it and a consistent way to do it, then I think that I guess overcomes one of the bottlenecks of the whole process, which is basically the putting the active sample on top of the slide. So that's something that we are interested in. So watch the space if you want to do spatial transcriptomics. But also I was very interested about the CellPlex system, the CMO system, which is a lipid tag. Goodness me, sorry. Can you hear me?
Yeah, it just switched slides, so it sounds like you're a bit further away, but we can still hear you quite well.
Sorry, it's very noisy now here. So no, so what I was saying is the CMO, so the classic lipid tagging of the sample, As you mentioned, 3, 4 is ideal for the workflow, for the sample prep load and everything. But I think that increases quite a lot of the capacity and decreases the price when you want to do a large number of samples. If you're able to multiplex them on the same day, then it's a one way to go. And the other option is as well that I was very interested is to hear more about the what is it called, the RNA Preservation Kit.
The fixed RNA, yeah.
Yeah, fixed RNA. So I think that also adds an extra capacity, overcoming another classic problem that is happening in this sort of sample prep and like logistics in terms of the sample, not logistics in terms of the molecular amplification and so on, but in the pre-steps getting into this. So I think that those are for me highlights. And you also mentioned something very, very interesting from my point of view, which is the pilot experiment. Doing a pilot experiment saves a lot of problems down the track, and having a kit that can quickly overview a little bit on your system that you also have available, because one of the limitations sometimes, although with the facility is easier because we can bulk orders and you don't need to buy a whole kit, you can do your own experiment, just one, and then you will be obviously using just the one. But having the access to do a pilot experiment where you can learn about your system in the actual way that you're going to see the big data. So if you have a mouse experiment or a specific tissue you want to do, you do a small pilot experiment with 1 or 2 samples, very easy to handle, and then get the data out. And from there, you upscale into the real experiment. That is absolutely is super valuable for you to learn what to expect from the system and how to handle your sample. Not everybody's working in PBMCs, so that is where the differences become quite critical. And it's not the same doing single cell RNA-seq in brain that doing in the liver or in spleen. And I guess that we are all learning about these differences, and so every project is different. So again, Yvonne mentioned that if anybody's interested in doing an experiment, reach out and we can have a chat on the what could be the best approach and the best option for that particular tissue, for that particular research question, in terms of multiplexing, how many, and depth and sequencing, and really all the aspects that Yvonne has been covering in the seminar, but ad hoc for the specific sample and questions that you have. So yeah, I don't know if I haven't been looking at the chat. Is there any question, anybody?
Couldn't see any.
Right. Look, this is again general overview of what's the best sort of considerations. Many of these considerations will apply to you. Many of them will not apply to you. So again, reach out, and then we can talk about the specifics of what is going to be the specifics for your project and for your question. That's the best way to start anything. So I don't know if you want to add. Thank you. So I think I don't know if you want to add anything else, Yvonne, at this stage. I think that you covered everything, but if you want to just give a final highlight to wrap it up.
Yeah. Again, please get in touch with David or myself. We can bring in the FAS team as well to, talk you specifically through some of the considerations that we want you to take when doing your experiment. There is a number of resources that we can point you to. I can send you a whole lot of things that cover these particular, how many samples, how much sequencing, what's the technical variation. We often get this question. There is some technical notes that we have on the website, and I know it is not necessarily all easy to find on our website. So I'm very happy to point you into the right direction for further reading, to get your head around what really is crucial for a single cell experiment and how can you best do it. We, as I mentioned, we're working with a lot of customers, and most likely someone is using a similar sample type to you, and we can share experiences there and success stories and best practices to make sure you are successful too in your experiment.
Excellent. Well, it's been very useful for me to go through all this process and get your opinions and your insights as well. And I would like to thank you for all these seminars, and I would thank everybody for being here today. And yeah, I'd like just to close this. I'm conscious of the time, close the seminar, and thank you, everybody. Please reach out if you have any questions. Thank you.
Yeah, thank you so much. Thanks, everyone. Thanks, David, for organizing this.
Thank you.
Bye, everyone.
Bye-bye.